Okay, continuing with the macromolecule of lipids, we're going to look specifically here at steroids and cholesterol and how they relate to the body. So starting first with steroids, they're composed of 4 rings of carbon, as we see them labeled here, A, B, C, and D. Examples are cholesterol, found in most animal cell membranes, and female and male sex hormones are also fall under the category of steroids, which is one of the 3 forms of lipids. Keep in mind, the other 2 are fats and phospholipids. So looking specifically at cholesterol here, it's a fatty substance found only in animal foods. It's also produced by our bodies. Small amounts are needed by the body for essential functions. So I want you to think that cholesterol is entirely a bad thing, because small amounts are still needed by the body. Where the issue comes in is where we have excessive amounts of cholesterol, and it can form this fatty plaque which sticks to the inside of our arteries. And this can cause heart disease. So we need our red blood cells to easily flow through our veins and our arteries. Here we have this build with these plaques. Now think about if we have blood flow going through here, everything is well and fine. However, if we restrict it down to here, we can have some serious issues. So we get heart attacks, various heart disease, even in extreme cases, death can occur. Now cholesterol, we have HDL, which is considered good cholesterol, and LDL, which is considered bad cholesterol. So LDL stands for low density lipoproteins, and it carries cholesterol around in the blood, and it can form those plaques I talked about in your arteries. As a result, it can increase the risk for heart disease, so negative things with the LDL pathway here. HDLs, though, are considered the good cholesterol. These are the high density lipoproteins, and these carry cholesterol to the liver and also out of the body for processing. So the liver can process it and synthesized by the liver, picks up cholesterol in the tissues, and can allow it to be excreted from the body. And this helps avoid plaques and heart disease. So when we talk about cholesterol, the broad term, please realize that there's low density and high density forms of cholesterol, and the high density are considered the beneficial ones for the body, while the low LDL, the low density lipoproteins, have negative effects. Now, triglyceride level in the blood is a fat that circulates in the blood and increases risk of heart disease. It looks like if you melted a butter stick with 4/5 gallon of water, produce a milky kind of looking liquid. So these triglycerides have that kind of milky look to them. Now, having none of them is not necessarily the goal. Under 150 milligrams per deciliter is the normal or level of low concern. Borderline high is 150 to 199. You can see high and very high levels. So having a certain amount is considered normal. But when we start getting into higher numbers, that's where we may get concerned, and the reason why we typically have blood work done to be able to assess these levels. Now, as a result, our normal blood levels of total cholesterol kind of shooting for that under 200. Borderline is over 200 to 239, and high risk is over 240. And that's milligrams per deciliter. We can break down that cholesterol instead of just that total. We can look at the high density and low density in the triglyceride level. So remember, the high densities are the beneficial ones. We want to see that number ideally 60 or above. LDL we want to be under 100, and triglycerides under 150 would be the goal. This is why we get blood work done. The reason why there might be these ranges or these goal amounts is we're trying to stay within that accepted kind of average or normal levels of distribution. Now, this isn't true for everyone, and they may be changing some of these levels, but it gives you just an idea of that total cholesterol under 200, but breaking it down further into the different types of cholesterol. So hopefully you get a better understanding for the difference between these two. Now, risk factors occur, so if you do have a family history of high low density lipoproteins, which is, negative cholesterol, that can be increased by if you're overweight and obesity. Your diet can also impact that. Your sedentary lifestyle, your family tree, your history, if you're a cigarette smoker, hypertension, diabetes type 2 can also contribute to this. And especially if you have a combination of these factors, can really increase the odds of you having a cholesterol problem. So if you have a family history and a sedentary lifestyle and an unhealthy diet, that can really help contribute to you developing this high cholesterol and negative effects seen by the body. Management, well, eating a healthy diet, exercising, keeping weight in a normal range, hypersensitive to diabetic, keep under control, don't smoke, and get your cholesterol checked at least once a year if you have risk factors. So just various good practices in general in both diet and health that help you be able to manage if you are at risk for cholesterol. Lastly, exercise. What we consider exercise can help boost your HDL or your good cholesterol. This reduces the risk of heart disease, helps control your weight, and helps control diabetes and hypertension. When I say exercise, this isn't just run up the stairs. This is exercise for at least 30 minutes daily. And you can choose from a variety of exercise that you help to enjoy, and you can keep it up. So don't pick something that you kind of have to do. Try to incorporate 30 minutes of some form of exercise daily that you might enjoy doing. So for some people that's running, for some people that could be swimming, that could be jogging. Find something that works for you, because exercise here is defined as 30 minutes of daily active work.